Hi, I'm Kashyap. My topic on today's presentation is microfluidic device with integrated micropump, which is an emerging technology in the present microelectronic industry. The main topics that I'm going to cover are: what are microfluidics, the history and evolution of microfluidics, the various components that are there in various microfluidic devices, micropumps, the main component of microfluidic devices, that is electro-osmotic pumping system, the various benefits of microfluidics, various applications of microfluidics, and the recent trends in microfluidic devices. What are microfluidics? Microfluidics is the science and technology of manipulating and controlling fluids, usually in the range of microliters to picoliters, in networks of channels with lowest dimensions from tens to hundreds of micrometers. A microfluidic device for nucleic acid analysis includes a monolithic semiconductor body, a microfluidic circuit, and a partially accommodation in the monolithic semiconductor body, and a micropump. Behavior of liquids in the microdomain differs greatly from macroscopic fluids in surface tension, laminar flow, fast thermal relaxation, and diffusion. A microfluidic device can be identified by the fact that it has one or more channels with at least one dimension less than 1 millimeter. Common microfluidics include whole blood samples, bacterial cell suspensions, protein or antibody solutions, and various buffers. Microfluidic devices can be used to obtain a variety of interesting measurements, including molecular diffusion coefficients, fluid viscosity, pH value of any liquid, chemical binding coefficients, and enzyme reaction kinetics. History of microfluidics. Microfluidics was developed in the 1980s mainly for the use in inkjet printers, DNA chips, lab-on-a-chip technology, micropropulsion, and microthermal technologies. Microfluidics is an interdisciplinary field with a wide variety of applications. It's a emerging technology in which it emerged in almost all the fields of sciences by the end of 2010. The main components of microfluidic devices. It consists of 6 components, namely microscale handling system, small volume transport system, subatmospheric pressure chamber system, sample loading and injection system, electro-osmotic pumping system, which is the very efficient one, which we will discuss in next slides, electro-pneumatic distributor system. Coming to microscale fluid handling system, a solution for conducting microscale reactions like digestion, separation, for efficiently transporting microliter to picoliter samples from a chip to an analytical device or a collection device. Advantages of this system are efficient sample transport, reduces the manipulations, reduces problems of sample carryover, savings, and less sampling time. Coming to sample loading and injection device, it is a solution for a universal interface device for transferring samples in series or parallel from sample container into channels of a multi-channel microfluidic device and into an analytical device that can be integrated into or separated from a microchip. The advantages of this device are: it uses standardized sample plates and variable sample volumes. It is reusable or a disposable device. Coming to electro-osmotic pumping system, this we will discuss elaborately in next slides. Coming to small volume transport system. It is a solution for moving small volumes of sample through the capillary channels or tubing of a microfluidic device, especially for a very long distances. The advantages of this transport system are minimal losses or minimum dilution of a sample, minimal cross contamination between samples, like minimal loss of sample to channel walls and washes inserted between samples. It is faster than for a sample changes. Multiple sample plugs injected at closely spaced intervals. Samples can be transported to long distances with high speed to any kind of devices. Next one is variable pressure delivery chamber. It is a solution for more efficient sample transfer from electrophoresis capillary or microchip to a mass spectrometer through an electrospray chamber by controlling pressure to allow fine control of sample flow rate from electrospray needle. The advantages of this chamber are minimal sample loss, lower evaporation of droplets, efficient dissolution, and minimal power supply source as it needed very less supply. What are micropumps? Micropumps are pumps with reduced dimensions and a microsystem which have preferably similar flow rates like the classical pumps. These pumps are of special interest in microfluidic research and have become available for industrial product integration in the recent years. Their miniaturized overall size, potential cost, and improved dosing accuracy compared to existing miniature pumps fuel the growing interest for this innovative kind of pumps. Coming to the important component of microfluidic device that we didn't discuss in the previous slides are electroosmotic pumping systems. It is a solution to control fluid dynamics in microfluidic device by using pumps to channel electroosmotic flow or pressurized flow in the device to perform sample transfer, gradient generation, or fraction collection or fraction deposition. The advantages of this type of pumping system are they are easily integrated into existing microchips. Its fabrication ensures high manufacturing and operating reproducibility, and it is a very simple design. Benefits of microfluidics. It has many functions that can be integrated into a single device. It has sensitivity, which is enhanced because of high surface to volume ratios. The small volumes of these microfluidics leads to fast reactions. It has a laminar flow which is very easy to control. There are lot many benefits of microfluidics, but these four are the major benefits because of which microfluidics is the present emerging technology in almost all the industries. Applications of microfluidics. The main applications of microfluidics that we can mention are the polymerase chain reaction, which is also known as PCR, is done fast using nanodroplets technology of microfluidics. It is done also for lab on a chip for bacterial culturing and testing. And some of the other biological applications of microfluidics are screening, genomics, diagnostics, and proteomics. To be elaborate on applications of microfluidics, I want to elaborate on fast PCR using nanodroplets technology. The recent advances in optical microfluidics have demonstrated the practical applications of how light can control a liquid sample in the same way as fluid circuitry. With laser heating operated on a droplet, a convenient reaction chamber unit for miniaturized assay systems, many optical fluidic control blocks have been simulated using this system. A laser has been used for manipulation and mixing of droplets, and a droplet valve sorter, fuser, or divider are all used in this system. The slide has proved to be an excellent method to mimic the classical microfluidic control on a planar surface. Many biochemical assays, however, require more than the fluid circuitry. For example, the polymerase chain reaction, one of the more prevalent biochemical assays, requires temperature cycling. One method commonly used for miniaturized high throughput droplet based PCR chips incorporates a heater or a temperature sensor inside the substrate, which is as shown in the figure. This approach requires complex design and fabrication steps. Miniaturized contract heating methods can be impacted by limited throughput and cross contamination if the sample is in direct contact with the heater. Infrared optical methods offer a simple non contact heating method in which the heating source can be independent and remote from the PCR mixture. While infrared heating for PCR can be performed with conventional light sources, a laser heating method can provide higher intensities and better power and wavelength control. Moreover, the same infrared laser that we used previously for droplet movement and mixing can also be used to drive the heating cycles of PCR on the platform. Another complementary advantage of the droplet approach is that the size of the droplet is easily matched to the short extension length of the infrared laser in water, thus normally following the trend of miniaturization while also optimizing the efficiency. Droplets as water in oil emulsions are needed in one of the most attractive candidates for that they are stable and can be easily generated and spatially arrayed on a solid substrate without crosstalk between neighboring droplets. In the second figure, the home built PCR chamber that holds the mineral oil and droplets at a constant temperature based on a conductive glass slide is shown. The figure that is shown in this slide shows the lab on a chip technology for bacterial culturing and testing, which is the emerging biological application of the present microfluidics. Recent trends in microfluidics. I have collected the information on the recent trends that happened in April 2016, in which the first one that happened on April 29th is that a research team from the Hong Kong Polytechnic University and Zhejiang University in China reported the integrating fiber optic glucose sensors into a microfluidic chip to create portable high performance low cost devices for measuring blood glucose levels in a patient to detect the diabetes in the blood of the particular patient. On April 8th, a team of 6 scientists, postdocs, and graduate students from biology and engineering labs won $300,000 for inventing a chip that sifts through a patient's blood to discover cancer far earlier than traditional tests can, which they did this in a competition hosted by the Koch Institute of Integrative Cancer Research to detect cancer in a particular patient. So, with this, we can say that these trends in microfluidics are mainly done in biological and human disease detection. So, concluding the topic, there is a rising demand for microfluidic chip miniaturization as it offers lower testing time and improved portability. Some of the factors that are leading to the growth of microfluidics includes rising incidences of lifestyle diseases, increase in research and development expenditure, healthcare spending, the growing stem cell and cancer research. Thank you for taking time and listening to my presentation. Have a good one.